Sometimes people think we're sitting in our ivory tower and we can't really explain what we're doing in layman's terms. And so I think some of that is the problem as well. It's not all on the policymakers. And so I think, us as scientists, learning how to communicate better, learning how to share our research better, not only with the policymakers, but people.
My guest today is Dr. Emily Cornelius-Ruhs. Thank you for joining me.
Yeah, I'm happy to be here.
You are a biologist at the U.S. Department of Agriculture, correct?
Correct, as of late.
Nice. Yeah. Maybe before we get into that, you could talk to me about your journey into the field of biology. How did that begin?
Yeah, so I think like most people who end up in biology and ecology especially, started on the pre-med track. So I always thought I wanted to be a doctor. I went to Michigan State as an undergraduate and quickly realized that I wasn't doing very well in the classes. And lots of people were doing well. And so in 2009, I studied abroad in Panama, where we were in the forest and we were catching poisonous dart frogs and doing some scuba diving and snorkeling. And I kind of had this epiphany, you can work on diseases and on animals and things in the health realm without actually having to be in the hospital. So I think I changed my major probably 2 more times, somewhere in there. But quickly after that, I started working in an avian disease and health lab at Michigan State University, doing all the grunt work. And I thought it was really exciting, even though I'm sure the professor was just like, oh gosh, I'm happy to pass off this project to an undergrad. But yeah, and then the rest is kind of history. Some of the work we were citing for that project with that professor ended up leading me to my master's program, which was in Georgia. I ended up, getting my master's degree with that same person that I was interested in, and then it kind of just snowballed from there. Oh, wow. I never stopped.
So where were you doing research in Panama exactly? Were you in like Smithsonian's, Barro, Colorado?
Yeah, so we visited there. We were in the nearby town of Gamboa, and it was really cool. It was also my first experience traveling really internationally. Oh, okay. So we did a field trip, like day trip to the Smithsonian, Barro, Colorado Island, walked around and hiked a little bit. And then from Gamboa, we went to Bocas del Toro, which was really where we did the snorkeling, and we did some island hopping for poisonous dart frogs. Mm-hmm. The TA, what a lucky TA, the TA on the trip was doing research on poisonous dart frogs for their dissertation. Nice. So we got to kind of, tag along, and it was really cool. That's really cool. Yeah.
And then, but for your dissertation for your PhD, mm-hmm. you switched again, or no?
Yeah, so when I was in Georgia doing my master's, I was working on migratory songbirds, trying to understand kind of the effects of migrating with like a parasite or pathogen. So I looked at avian malaria parasites, they're blood parasites, and really wanted to stay there for my PhD. But my partner got a job up in Wisconsin, and so I started to look north and was actually about to kind of call it quits and just look for a job when the PI, Bill Karasov at the University of Wisconsin, posted this position that seemed perfect. It was immune function of a residential bird species, the black-capped chickadee. And after speaking with them, it kind of seemed like I could take it wherever I wanted it to go. And so, I ended up there. It was kind of kismet timing.
Nice.
Yeah.
Was there any kind of singular moment that stands out that made you want to become a biologist, other than the pre-med thing?
Yeah. I think for whatever reason, I kind of always wanted to be a veterinarian. And to be honest, to this day, I'm like, man, should I have become a veterinarian? In some regards, it would have been easier. And I think in terms of like the health and disease effects, I remember in high school, my grandma actually fell down the stairs and had like this, subdermal hematoma. And I remember it being kind of like a critical mark for me, where I was like, at that time at least, it was like, I'm going to be a neurologist. I want to do that. And I spent a lot of time working for neurologists and neurosurgeons at hospitals and loved it. I mean, I think you can, it's just like the fascination and the curiosity of it all. Mm-hmm. But and then quickly switched when I was like, I can be outside in the rainforest. Yeah, right. But I mean, still answer these questions about health and disease, right?
Yeah.
So, yeah.
Right. Yeah, I mean, One Health, and on the heels of COVID, obviously it's a pretty important field of research.
Yeah, I mean, I think so. I think there's a lot of different ways you can take it. And yet people are starting to recognize that even more after COVID, which is nice. It does seem like there's a lot more funding and some positions that are opening up that kind of explore those relationships. And so I kind of, lump my research in that One Health arena. I've always had an interest, probably from early on being interested in the human side of things. I've always had an interest in public health. And so I think it's a good realm to be in. I think there's a lot of questions to be answered. Mm-hmm. So, as you know,
And always new pathogens, right?
Always new pathogens. And even the pathogens that have existed for a long time, we still don't know a lot about them. So,
mm-hmm. What are some of the challenges you faced when you first started your career?
Yeah, that's a great one. I mean, a lot of it was imposter syndrome, which you hear from a lot of people, especially women in science. I had a really great time with my master's. I had great advisors. It was really felt supported at the University of Georgia. And that was pretty much true at University of Wisconsin, Madison too. I had a great PI who kind of let me take things in directions that I wanted to take them and was there. Not always to catch me if I fell. I think that's a part of the learning experience, but was on my team. I think I started to have some challenges after that in terms of postdocs and you get into this realm of how does authorship work and who owns what and how do I do this? And so I had a lot of challenges post PhD trying to find where I fit in in the world of science. But again, as soon as I kind of moved to the University of Chicago and we worked out the kinks of being in a new lab, I mean, I've loved it. And the Field Museum has been an amazing place to work. I've really enjoyed it. The people there are very kind. And so I think it's allowed me to kind of spread my wings. And so, I mean, a lot of the challenges were just learning how to work with people in different groups. And when you're a postdoc, that changes like every year or two, which is, you kind of feel like you're starting over again. So that's kind of been the biggest challenge.
Yeah. I think a lot of people suffer from the imposter syndrome at some point in their lives, myself included.
For sure. Yeah, and what's really stuck with me is that my PhD advisor is well known in the field of digestive physiology, something that I know very little about. And I remember him saying to me once, when I'm in a room of physiologists, I tell them I'm an ecologist. And when I'm with ecologists, I tell them I'm a physiologist, so they don't expect anything from me. I'm like, man, if this guy who's like at the pinnacle of his career and everybody knows who he is feels this too, like, on the one hand, shoot, I'm going to be feeling this all my life. And on the other hand, I'm like, okay, it's not just me,
Right.
Yeah.
What gives you the motivation to keep going during hard times, or when you go through hard times?
Yeah. Ooh, that's a really good question. And I saw that. I was like, how do I answer this? I mean, I think, you and I spoke about this a little bit in the beginning, right? It's like when you're really passionate about the questions, you're very passionate about biology, even though it's not your main career. Every time there's like a new data set or something comes through the pipeline and it kind of worked, or you can explore it, like all those bad moments go away. And so I think that's when I've struggled before with either, PIs or collaborators, I'm like, man, but I'm still really interested in the data. I'm still really interested in the project. And I just wish sometimes the people would go away.
Yeah.
And I think very recently, the projects that I've built trying to become an independent scientist, I've built them because I get to work with, I get to work with cool people. And I've decided that I just want to work with the people that I like and as much as possible to do these fun research projects and answer some cool questions.
Yeah. Could we talk a little bit about your current position with the U.S. Department of Agriculture?
Sure. Yeah.
What will you be studying in this new position?
Yeah, so I came on December 1st as a biological scientist, so I'm very much still getting my feet under me and doing onboarding. But I have been tasked with trying to understand the high pathogen avian influenza, HPAI, and kind of how it's spreading, where it's going. There's already a team in place there that has been working on it for a really long time. But in particular, they've asked me to kind of help with the dairy cattle. So dairy cattle have been infected with it in pretty high rates in across the country, but really in California right now. And so, yeah, I'm trying to do some modeling and understand where it's going next with, dairy cattle movement and things like that. And then the other thing that I'm really excited about is to dive into some wild bird surveillance. And that's like, as of last week, I was asked to help on that, so I can't really expand. But they also do surveillance for wild birds. And so I'm excited to do that because it seems a little bit more up my background and alley. Yeah.
So you can't talk about the bald eagles stuff?
I can talk about the bald eagles.
Oh,
yeah.
okay.
So that is totally separate.
That's not with this?
That's not with the USDA. They've been kind, and they're going to let me kind of keep on with that, but no, it's not.
Well, could you talk a little bit about your bald eagle research?
Yeah, I would love to talk about the bald eagle research. So in 2023, just a couple years ago, a collaborator of ours, and I'll say ours because I have, the other co-PI on this grant is Gavin Dainert, who's a research scientist at Wisconsin Sea Grant. So we knew the leader of the Bald Eagle Project just as a collaborator and kind of like a contact for many years. And he called in 2023 and said, hey, I've always wanted to know what's going on in these eagles beyond what we've kind of been doing with them. So we were like, heck yeah, I would love to work with bald eagles. Yeah. And so the story with that project is it's been kind of like a biomonitoring project for many years. It's changed hands a few times, but it's always been with the Wisconsin DNR. And so Sean, who's the leader on the project, is the one who called us. And so for many years, like decades, they've been pretty much every year taking samples from nestling bald eagles, so before they can fly, and monitoring them for heavy metals, PCBs, PBDs, and now PFAS. And so when Sean called, we were like, yeah, we can add kind of like a standard veterinary exam to the bald eagles, and we can try and actually get at the question of what is, being contaminated with these toxicants mean for their health. So we started that in 2023, and just like everything, it snowballed. So very recently, we received 2 major federal grants to continue that work. So we have funding for the next 4 or 5 years to keep monitoring populations, and we're expanding this year to look at, the entire Wisconsin River up and down, hopefully, and then following year will be the entire Mississippi River. And then I think it's the St. Croix River, which is closer to Minnesota. And so, yeah, that's kind of the background of the project, but we're adding a health flair onto what they've done before.
It's interesting with the bald eagles too, right? Because the DDT thing, populations crashed, we banned it, or the government banned it. The populations came back.
Right.
Have you noticed so far that PFAS have caused a population decline in bald eagles?
So, to my knowledge, no. I think it's kind of lying under the surface. So we have some historic data from the bald eagles, obviously, from those past samples. And the challenge with kind of comparing what's happened in the past with what's happening now is that the panel for PFAS testing keeps changing. And so I think so previously there were maybe like 16 or 17 analytes in the panel. We send the samples out to the Wisconsin State Lab of Hygiene, and now there's 33. And so now in this like 33 panel test, we detect PFAS in every single sample of bald eagles that we test, different levels, obviously, depending on the analyte you're looking at. But I don't necessarily think it's causing mortality. I think what we're talking about with that is that it's morbidity effects. And I can get into that more. But I don't think it kills them directly. I think there might be kind of like multiple things are happening under the surface, and then it could lead to effects that It'll
be interesting to see if PFAS, these forever chemicals, will be banned like DDT was back in the day. Yeah,
so and I'm not a toxicologist, but my understanding is that the US has kind of voluntarily started to phase out or ban some of these. I know like PFOS and PFOA are the 2 big ones that you hear of a lot. But the problem is, as I just mentioned, there's 33 in the panel that we detect, but there's actually like greater than 14,000 types of these analytes.
14,000.
There's a lot of them, yeah. And I don't even know if we know like really what all of these mean and what they are.
Yeah.
And so banning is a little bit complicated because there are reasons why these were created, right? Like they have some good effects. There's a reason why we didn't want our eggs to stick on our pans in the morning, and you know, more broadly why they're in firefighting foams. And so I think with PFAS, it's hard because, you know, PFOS is one of the big ones. It occurs in levels anywhere from like 0, obviously, in our eagles all the way up to 250 nanograms per milliliter. But something like, you know, I like to call it PFUNDA, but it's PFUNDA is in levels that are like, you know, 2 to 10. And so to ban it across the board is a hard thing because, you know, different levels have such different toxicities, right?
Yeah. I had no idea there were so many within that PFAS grouping.
Yeah. And again, I don't know all there is to know. That's why Gavin is on the project. He's the toxicologist. But yeah, there's a lot of them. And so, you know, we're picking up the 33 that are in the panel, but there's other ones that are out there that I'm sure that they have in their system. We just don't have a test yet for it. I'm sure it'll change maybe even the next 5 years, see what's in that panel.
Right.
Yep.
So it's too early to tell the way the PFAS are affecting bald eagle health then. Right?
So we have some preliminary results. So the first year that we worked with the Wisconsin DNR group, and it's many people who kind of either volunteer their time or, you know, do it because they love it. And we did basic veterinary assessments, you know, like you take your dog or cat. We're going to do complete white blood cell counts. We looked for natural antibody levels. We looked for something called complement lysis. So the complement system kind of bridges the innate and adaptive systems and signals, you know, things to work the way they should. And what we found is actually for some subtypes, some analytes of PFAS, we're seeing decreased white blood cell counts, which could indicate that these bald eagles are immunocompromised. To a certain extent, we're seeing decreased antibodies, which kind of matches the literature because most of what we know about PFAS are in humans, and a lot of it has been looking at how people respond to vaccines. So that kind of tracks. And the results are kind of still coming in, but one of the other cool things we did is you can take a blood sample from an eagle and you can keep it alive, if you will. You can keep it warm, and you can put it in a tube and challenge it. So you're not doing anything to the eagle itself. We're kind of doing the standard blood sample that we typically do. But we challenged the blood with a bacterial challenge and a viral challenge. And the viral results aren't back yet. They're being sequenced. But what we see is that gene expression in the bald eagles from areas where they're highly contaminated actually seems a lot lower. And so if you're saying then, this is kind of why I said I don't think it's killing them, but it could have a long-term effect. If you're saying eagles that are highly contaminated have a lowered gene expression to something like inflammation or antiviral, and then they get something like high path avian influenza, they might not survive as well. And there's a lot of factors going on, but that's kind of what we're seeing. And so I think it does play a big role in how they might respond to some other sort of challenge in the future.
Well, one of your other areas of research has been on bats. Could you talk a little bit about the research you've done with bats?
Yeah, so as I mentioned, my background's in birds. I like to joke that I'm a flying vertebrate immunologist. And if you know anything about immunology, or you're kind of in this world, I've always wanted to work on bats because they're just cool. We seem to think that because of the evolution of flight in bats, and some other things too, right? Like they have high metabolic rates because they have the highest sustained powered flight of any animal, to my knowledge. And despite that, they are actually incredibly long-lived too. And so typically what you see with something that has that high metabolic rate is they actually have really high cellular damage. but they're living longer. So you kind of say, what's going on? And there have been tons of papers describing this and why they're so like immunologically cool. But in those bats, then you see things like, dampened inflammatory responses and increased DNA repair mechanisms to kind of counteract some of those effects of flight. And that's the hypothesis is that's why they're able to be such competent hosts for many of these viruses. And so from a purely, nerdy immunology perspective, I just wanted to work on bat immune function things and also kind of dive into the realm of zoonotic pathogens, so things that can infect, move from animal to human. And so, so I had one paper that was kind of just a literature search, trying to understand scaling or the relationship between body mass and immune cells in bats. But then more recently have been working with Kara Brook at the University of Chicago in Madagascar to try and understand the ecological drivers of some of the disease patterns that we're seeing in Madagascar. So, yep.
Whereabouts in Madagascar?
Yeah, so I'm definitely more on the lab side of things, but they have sites kind of all across the country from the north to the middle to a little bit in the south. So there are sites near Antananarivo, not too far away, but in Madagascar, it's still quite a drive.
Yeah. Right.
Yeah, and then there's sites all the way up in Ankarana, which is all the way up in the north. So I've had the pleasure to be in Maramiza and Engavkeli, which is just to the east of Antananarivo, the capital city. I've also gone to Analambutica, which was beautiful, and that's a little bit northeast, I think, of Antananarivo. And that was like this beautiful valley where there were Tropis fruit bats, and we got to camp and spend a week, and it was really fun.
Yeah, it's a special place.
It is a special place. I think I like it. I mean, I love it more and more every time I go there. It's just really cool. Yeah.
Could you talk a little bit about the trainings that you've done in Madagascar then?
Yeah, the course.
Yeah, the course.
So together with Kara and her grad students, each year they put on ecological and epidemiological modeling in Madagascar, E2M2. And that's about a 9-day workshop where we train students pretty much primarily from Madagascar. They're PhD students. Many of them are from Antananarivo and some are from other universities across the country. But we train them in statistical skills, essentially. So anywhere from, simple plotting and making figures in R all the way to linear models. And then a lot of people in the group do more with modeling than I do. So we kind of move up to a SIR model, so a susceptible, infected, recovered model, and show them how to do it in R. We do some fun tutorials, but they also bring their research questions. We kind of ask them, what stats do you think you would use? And how can you see, some of the information we've given you? How can that help you essentially along their way? And the point of it is to help students get to be able to write a paper and have the statistical methods they need, but also to think about their research questions in terms of, what's the data I need? And is this a statistical question? Is it more of a mechanistic modeling question? And so it's fun. There's usually 25 students that show up and they go.
And that's annual? Pretty
much annually. Kara has done it since maybe 2015 or so. It's been going on, and it obviously paused for COVID. There was one year we instead pivoted, and National Geographic gave some money to do coding for conservation, which was more of a year-long mentorship, and it was all remote. But everyone was paired with an individual student, and we tried to help them bring, data and a question to a final publication. So, nice. Yeah.
Could you talk a little bit about that paper you wrote regarding bats, body size, and immune cell proportions?
Sure.
It's pretty interesting.
Yeah, so that was really my first true postdoc out of my PhD. It was at the University of South Florida. So we were interested in how immune cells, and that project in general was also looking at how immune networks scale with body size. And so there's this long history of things scaling, or allometry, where, there's a predictable relationship between something like, small intestine size and the body size of the individual. There's, it's true of, metabolic rate and all kinds of things. And so there is a really awesome database. It is behind a paywall, but it's available to people to use, and it's called Species 360. Every zoo that's accredited in the country, to my knowledge, enters their data there from their standard veterinary assessments. So we had this really nice database of things from birds and other mammals. There's not a lot of bats in zoos. And so then, together with Dan Becker, who's a professor at University of Oklahoma, but double check me on that. We gleaned data from the literature and pulled out every single immune cell proportion we could. So these, again, are kind of like your complete white blood cell counts that you would get, or your dog or cat would get. And so in a previous paper, we showed that birds actually have way more cells for their body size than mammals of the same size. And that kind of happens after a certain point, like a small mammal, like a mouse and a maybe zebrafinch, have kind of the same, but when you scale up and you're looking at an emu and a coyote, birds have somewhere like 2.2 times more circulating cells than mammals.
And why do we think that is?
So that's a great question. We kind of argued that it's possible that these birds are exposing themselves to more things just through flight. It's a little unclear to me still. I was only in that position for a couple years, so I think they're still working on that project. But there was this really interesting pattern where if you got over log scale of 2 for your body size, there was like this big shift where things started to diverge and be a lot different. And so it's a little unclear, but the thought with the bats then was, well, mammals are lower than birds, but bats are somewhere, they're mammals, but they fly. And so the unfortunate part is, you don't have bats that don't fly, and bats are the only mammals that do fly. And so it's a little hard to make that comparison. But when we then compared mammals to birds, terrestrial mammals to birds to bats, the birds were obviously still different from the mammals, but the bats weren't different from either. And so what's going on there, I'm not really sure. There's also not a lot of literature or data out there on bat immune cell proportions. And so to be able to answer that question better, you'd really need to get more data. I think we had data from 60 species, and there's more than 1,400 species of bats, and so it'd be really cool to be able to expand that further with a larger range of body sizes.
And the bats data that you did have, that was all from bats in captivity then, or no?
No, a lot of it was from wild bats, but kind of like a mix of both. And so there are some issues there too. There was another project that was led by the PI down there where we looked at captive and wild animals, and we didn't really see any differences. And so, yeah, a little unknown, but I think that's a push for, if you can get a small blood sample from something that you're a bat or an animal in general that you're, measuring the wild, it can be really useful later to keep. And blood smears are pretty easy. And so it can give us a lot of data and information on what's going on there. Yeah.
With bat research, have you done any work with white-lined bats?
I haven't.
Okay.
No, the only bats I've worked with are the ones in Madagascar.
All right.
Yeah, sorry.
Fine.
I would love to get something going in the United States. It'd be really fun. Yeah.
What about, have you done any work with white nose syndrome in bats?
So I haven't, but I know a lot of people who do. There's some cool stuff going on for obvious reasons. But no, I've never had the opportunity to work on that. But yeah, lots of people are.
Yeah, it's an important one, right?
It's an important one, yeah. And I know there's, some cool stuff in terms of using UV light to get rid of the white nose fungus, and there's also some cool stuff. I'm not sure how much further they've taken it, where the Bat Conservation International has tried to do this Fat Bat project, where they lure bugs in to just outside the cave during the swarming season, and they try and fatten up the bats before they go in, because if they're fatter going in, they're more likely to survive this whole, white nose and arousing too much. So yeah, there's some cool stuff, but I've never worked on it.
Yeah. Another paper that I came across that you wrote was regarding Arctic animals and migration changes due to climate change. Could you expand on that a little bit for us?
Yeah, so for many years after my PhD, I was the associate editor for a journal called Animal Migration, which unfortunately no longer exists. But it was a really fun process and allowed me to work with a lot of people that I wouldn't have otherwise. And so we would put on these special issues. And this one in particular was, animal migration in a changing world, essentially, or, how does climate change affect animal migration? And so together with Sarah, we wrote this paper that was really more of a review. She works on MoveBank, which is a really cool website and database where people can input their information about the different species they're working on. And so that was kind of the introductory article to a lot of other really cool articles, not only describing migration, but also just sort of the distribution of these migratory animals at different periods in their life and how they're changing with climate change. So yeah, we did 2 or 3 special issues while I was the associate editor there. Another one was stress on animal migrations, which was with Andy Davis, who's really the stress guru, especially with birds. And so it was a really fun process. I feel like I got to expand what I knew, because I definitely don't work in the Arctic, and I don't work on migratory animals a ton. And so yeah, that was a fun process.
What happened to the journal, if you don't mind me asking?
Yeah, I can ask. It got hacked. It got cut, unfortunately. It was part of a larger umbrella, and it wasn't, these sort of special issues were our way to try and bring in cool research and broaden. But I think because it was free to publish, the journal, the larger umbrella, kind of was just like, we're not supporting you anymore, which was too bad.
It was too bad.
Yeah.
The chickadee paper that you wrote was interesting. Could you talk a little bit, chickadees faced with unpredictable food increase fat reserves, but certain components of their immune system decline. And when I think about that, it would make sense that they would fatten up the reserves going into winter. But could you talk about what components of their immune system decline? Is that in response to increased fat reserves? Yeah,
we think so. So the paper you're referring to was really the first paper out of my PhD. And so what we did, that study was experimental. So we caught the chickadees in a local forest, and this was all done in collaboration with researchers at the University of Quebec at Rimouski, which is 3 hours east of Quebec City. It's pretty far. And so we experimentally manipulated their food, and it was pretty cool because it was on an individual level. So like I measured, I measured both of our food intakes for a week, and then I said, well, you need this many calories, I need this many calories. Let's cut that by 40%, and we'll give you 60% of what you need on random days. So you don't know when you're going to get a Big Mac versus when you're getting a salad, basically. And so, there's been kind of these 2 camps in the literature. Small birds tend to increase their body mass, which the chickadees did, as you already mentioned. It's kind of like a hedging your bets situation where I don't know if I'm getting food the next day, I'm going to kind of store up some extra fat. And other animals, for obvious reasons, decline. It'd be like if we went on a diet. And so what I found is that when they were increasing their body mass, and more specifically their reserves, like their fat reserves, things like response to external stimuli declined. And so we also challenged them with lipopolysaccharide, which is a bacteria. It doesn't hurt them at all, but it mimics like if they were to get the flu. And so when they were, the more fat they gained through this process, the lower their immune response was. So there was this really clear trade-off between kind of the inflammatory response and fat gain. And then one of my dreams in the future is to dive into that further, because we actually don't know how they're able to gain fat when they're given less food. They didn't eat more food on the days where we gave them more food. So They weren't like, eating a salad and eating 4 hamburgers the next day. They were just basically eating the same amount each time. And so something else, they must have downregulated something like, doing some energy saving at night or lowering their basal metabolic rate, which these guys are known to do really well. And so that's what I want to dig into again. Like, how are they able to actually do that?
Right.
Yeah.
What's the mechanism?
Yeah. Yeah.
Oh, this is well, it's chickadees as well, but I had come across a paper regarding black-capped chickadees where their brains grow by 30% in the fall. Did you see that paper as well? Or?
I don't know. If it's a recent one, probably not so much, but I think chickadees are really known and used for these types of studies because they have crazy spatial memory. And so people study their hippocampus a lot. And there's a researcher out in kind of like the Reno area who's been doing a lot with their spatial memory for many years. I've never done anything with sort of their brain dynamics, but just in general, I think these chickadees are pretty phenomenal because they're at most a 12-gram, 14-gram bird. 14-gram would be a pretty fat chickadee. And so to be able to like be so phenotypically flexible with so many of these scenarios that they undergo is pretty cool. Yeah.
Yeah.
So
like a 30% increase is pretty amazing.
Yeah. Yeah.
And then it shrinks back down, or it expands and then it shrinks back down, I think.
So I'm not sure I've read that paper, but I can see that. I mean, when we were talking about the fat mass, they increased their fat mass by 300%. And so this is a chubby little chickadee, and they did it in a matter of 5 days. And the other things that we've done with chickadees too is like sudden cold snaps in the lab, and they can very quickly adjust their internal muscle structure to deal with those like quick cold snaps. And so they can do things pretty fast. They're pretty amazing little guys. Yeah.
Well, switching gears a little bit, but something that these are some of the questions I didn't get to you in advance.
No worries.
Has there been a work of fiction, a book that has helped you on your journey in biology?
Yeah, that's a great question. I love reading. I'm a big reader. Recently I'm reading Fourth Wing, which is all about dragons, so it has nothing to do with biology. I tend to bounce between fun murder mysteries and then I try and read something that'll make me learn in the process. Very recently, I read a book on Chagas disease, which was really cool. It was a little bit more from a journalistic perspective. And so this woman who's from Colombia and had family members in Colombia finally dove into, why do people from this area tend to get Chagas disease so much? So I like to intersperse those, but I think one of my favorite books was David Quammen's Spillover, which is maybe makes sense.
But that's nonfiction, right?
Yeah, that's kind of. I mean, he went out and explored some of these regions and talked about these diseases, and it came out maybe in 2012 or so. And so to read his last chapter again, which is called Disease X, after the pandemic happened, when everyone's talking about Disease X and what's going to happen next, and can this happen again, it's cool. It's cool to go back and read those books that pseudo predicted what could potentially happen and then see how it pairs. I also really enjoyed, I'll blank on his name, but he wrote the books like The Big Short and things like that. But he had a book called The Premonition, which at first I was like, I don't know if I want to read this. It's about COVID. I just dealt with COVID. It's still going on. But he took it from a, how did the government respond and what did we do wrong and what are all these things that us as everyday Americans might not have known about. And so that was cool too, because I started to also see people that I recognize in the field of disease and disease ecology in that book and kind of like how they helped during the pandemic and all that. So kind of nonfiction, but mostly fiction. Yeah. He would call it nonfiction probably.
Yeah. What has been the most surprising thing that you've learned in your research on birds and bats?
That's a good question. We don't know a lot,
Yeah.
I think, and that kind of speaks a little bit to the disease side of things a little bit more. I'm just starting to jump into high-path avian influenza and hit the ground running, and there's a lot we don't know. And that's true. I mean, that was true of COVID, obviously, and that's very true of the viruses that we work with in Madagascar. We're still trying to understand when they occur, how they get infected, how they deal with it. And so I think overall it's just there's a lot to explore still.
Yeah.
And so I think sometimes, me, especially when I was working on the chickadees, while I loved it and it was super fun, I was kind of like, why does this matter? Why am I doing this? And that sort of imposter side of my research has gone away a little bit the more and more I move towards things like, zoonotic diseases, or especially high-path avian influenza, where it has this really applied focus. But I think there's a lot we need to, I think there's a lot to learn still.
Right.
Yeah.
Yeah, I think that's what makes biology exciting at the same time,
It does.
right? Is that there's so much we need to figure out.
Yeah. Well, and I mean, who knew trying to understand the expansion of the brain of a chickadee would mean anything? But it does. It has implications for humans. Not that, we can circle back to One Health. I don't think it always matters to tie it back to humans. But I am enjoying the fact that my research is moving towards a little bit of the public health side. The bald eagle work, which we talked about, has so many implications for the communities that also feed on the same things nearby. And so there's a lot of applied things that are important to try and understand for the animals, but also the humans that live in those same regions, right? So.
Yeah. Do you have any memorable fieldwork experiences that you'd like to share? I know you talked a little bit about the stuff in Panama, but has there been something else that stands out?
A couple things. I mean, so the funny thing about the Panama trip was, I was a sophomore in college, and when we got to Bocas del Toro, which is really a beach town, they were training us on how to snorkel, which I had been a couple times before, but I also knew we could go to the forest and we could do, this poisonous dart frog research. And as soon as I got into the water, you're walking out on the beach and there's seaweed. You have to cross over the seaweed bed to get to the fish that you're trying to look at. And I immediately had a panic attack.
From the seaweed?
Do not like things touching me. This is why to this day I can't really snorkel. And so I must have started flailing because my professor, who was probably 65 years old at the time, carried me like a baby out of the water and dropped me on the sand. And I'm, just sheer adrenaline at this point. And I don't even know what he did afterwards. He was probably like this kid, and he went back in the water. But I got stung by my first fire coral, and it hurt really bad.
Yeah.
So that's a really memorable one. I mean, with the chickadee research, it was actually one of the last years I went up there to do research. It was for the cold snap research we were doing. We had to catch a bunch of chickadees and bring them into the lab again. And I had a great team of undergrads from the university that were there who were helping me, and for hours. And this is we would catch these guys in the winter. And so it was cold, it was snowy, they're little tiny birds, you're freezing your butt off. I had three undergrad guys who were with me, not a single chickadee the entire time they were there. It was hours. They were like, sorry, we gotta go to class, we gotta leave. And I was like, cool, I'll take down the net. I've got this covered. And I go and there's 50 chickadees in the net as soon as they leave. And I, again, remember the adrenaline of oh my God, I'm taking so long, these chickadees, I want them to survive, and I got them all out. But I was like, oh man, it's time to close it up. It's time to go back. So there's fun little things like that. Yeah. But field research is why I think everybody does this, really. It's just fun.
Most people,
Yeah.
But there are people that like the lab work,
That's true. That's true. And I like the lab work. It's different. Yeah.
Where can listeners learn more about your work? And
I can say this is that for the, I obviously have a website. People can go check things out. So do all of the PIs I've worked for. But for the Eagle project, I'm excited because the grants that we got obviously have an outreach component. And so we're building a website to try and have, listeners and people who are fond of bald eagles or people who want to contribute That'll be coming up soon, where they can read more about the project, they can see who the team is. This is especially important for, we go to a lot of private land and private homeowners, and so they can kind of see who we are before we go on their property. I'm hoping in the future we can do almost like little report cards for the eagles that we're catching, especially on these private properties, where they can say like, your eagle is WI204, log on to the website, you can get the results, we can put it into the greater context of what the PFAS scene looks like in Wisconsin, and you can kind of try and understand how your eagle is doing compared to the other ones. It's a long time, there's a lot of work to be done to set that groundwork, but in the future there will be a website for that. It's GreatLakesEagleHealth.com.
GreatLakesEagleHealth.com.
Eaglehealth.com, yeah.
How can science and research be combined with policy changes?
Oh, that's a big one.
Yeah, it is.
Yeah, that's a really good question. I think it goes both ways. I think policymakers, lawmakers need to believe scientists. I think scientists need to get better at explaining their science to the general public. There's a lot of push to help scientists be better communicators. Sometimes people think we're sitting in our ivory tower and we can't really explain what we're doing in layman's terms. And so I think some of that is the problem as well. It's not all on the policymakers. And so I think, us as scientists, learning how to communicate better, learning how to share our research better, not only with the policymakers, but people across the state, because, they vote. But then I'm always amazed at policymakers not believing scientists. And some of that's for obvious reasons, but I think there has to be a little bit of trust across the board. So I think that's a pretty good starting spot.
Yeah. What is something you've learned from your research on birds and bats that has informed something in your own life?
Yeah, wow. Something that I've learned from birds and bats that have informed my own life. We don't know anything. No, I'm just...
It's valid.
No,
right?
That's a really good question. I don't know if I have a good answer for that.
It's okay if you don't.
Yeah. To be resilient, I don't know. I mean, these I've worked with a lot of small things. The chickadees are incredibly small. Bald eagles, not so much, but they're incredibly resilient, which is pretty impressive. I don't know if that informs my own life, but it always amazes me that, these eagles can be highly contaminated and they're still feeding their chicks and nestlings. And so I guess there is some parity to everyday life. But I think it's interesting. It's not a field I've ever gotten into, but the comparisons between, like how we're all pretty similar. We all have these family structures and social structures. And so I think there's a lot of comparisons that can be made there. I don't know if I can come up with them on the fly.
Yeah. This idea that the interconnectedness of life, right?
Yeah, and I think you actually, it brings up a good point. You've asked earlier some of my readings and things that I like. I love Ed Young's stuff. He always has really good insight. If you've ever read any of his books,
I have not. No.
And I think he's moved some of his books to be kind of more kid friendly too for certain ones. But yeah, you should check him out. He has good writings about kind of the ties between the human and the animal world, but beyond that too. He writes for The Atlantic, but has a number of books. Yeah.
I'll check it out.
Yeah.
How has your work studying bats and birds impacted your humanity? I think we kind of went over that. Unless you have something for that.
I don't know if it's the birds and the bats specifically, but you've been to Madagascar, you mentioned. It's been really, I think just the research in general has opened my eyes to all these different cultures, which has also been really cool. With the bald eagle work and some of the PFAS work, we get to work with the tribal cultures in Wisconsin. The Madagascar work, I love the country, don't get me wrong. It's warm and the food's great and the animals are cool, but the people are amazing. And so all of those connections that you build from doing field work and going to do these research projects, I love building the connections with people too. So yeah, there's just amazing people to help you in your research.
If you had one piece of advice to someone entering the world of biology, what would that piece of advice be?
Yeah, I would say keep going. And that's true, especially as a woman in science. especially as a woman in science I have kids. I think you can do it. You can do it. And that's easier said because I have a very supportive husband and family. But keep going. Don't let the jerks and the imposter syndrome get you down. Because if you're interested in it, there are people who will support you, and there are places you can look for help. And I think there's a lot of women who will help you along the way to get there. So I think that's my biggest piece of advice, is you can do it.
Yeah. That's a great way to end.
Yeah, perfect.
Thank you, Emily. I appreciate the time.
Yeah, definitely.
Thank you.
Happy to do it.